Thank you. So good morning, everyone. Welcome to the Last Drop Water Researchers Speaker Series. This speaker series is brought to you by the University of Manitoba's Sustainable Development Goal 6 Working Group. And in 2018, to give you some background on this group, the University of Manitoba was named the United Nations Academic Impact Hub for Sustainable Development Goal 6, which is clean water and sanitation. We are the only academic institution who holds this UNAI designation in North America. And the SDG 6 working group at the U of M brings together established and emerging leaders who have a shared interest in freshwater sustainability and to work collaboratively with national and global partners to address the issues related to climate change impacts on freshwater systems at local, regional, and national scales. Our members include researchers from the Clayton H. Riddle Faculty of Environment, Earth, and Resources, the Faculty of Agriculture and Food Sciences, the Faculty of Arts, and the Faculty of Science. And we have collaborators from the Center of Advancement of Teaching and Learning and the Office of Sustainability. So our expertise spans multiple disciplines across the humanities, social sciences, and STEM fields. And my name is Claire Herbert. I work within the Center for Earth Observation Science, which is a department within a center within the Clayton H. Riddle Faculty of Environment, Earth, and Resources. And I'm a data manager for a data repository, and I also a program manager and biologist for the Manitoba Great Lakes program. And I co-chair the SDG 6 UNAI working group with Dr. Nicole Wilson. So in this virtual format, we are all coming from different locations. Feel free to put your territorial acknowledgement in the chat. And I want to acknowledge that I am located in Winnipeg, Manitoba, at the University of Manitoba, on the original lands of the Anishinaabe, Cree, Ojibwe, Dakota, and Dene peoples, and in the homeland of the Métis Nation. And in the theme of today's panel, I also want to acknowledge that my drinking water comes from Shoal Lake through the Winnipeg Aqueduct, which was constructed in 1919 at the expense of Shoal Lake 40 First Nations water security. So today is the first talk in our series for this 2023-2024 season, and it's by Dr. Ayush Kumar. So thank you very much, Ayush. So some logistics. We've enabled the closed captioning function, so feel free to turn it on. As you noted when you were let into this room, I believe, we will be recording this event and it will be on our U of M Sustainability YouTube channel after the talk, and the link will be put in the chat, and it'll also be available on our SDG 6 website. The presentation will be around 30 minutes, and then there will be a Q&A period at the end. So feel free to put questions in the chat. And there will be someone monitoring them. And if you want to learn more about our group or see YouTube videos from last year's presentations as well as this year's, you can visit our website, which will also be put in the chat, or you can just search U Manitoba SDG, and you should be able to find us. So now I'm going to introduce Dr. Ayush Kumar, who is going to be talking today about microbial quality of drinking water from First Nation communities in Manitoba and an urgent need to upgrade the water distribution system. And Dr. Kumar's research group has been actively studying the mechanisms of multidrug resistance in gram-negative bacteria. They have characterized multidrug efflux systems in various organisms, such as Acinetobacter, Pseudomonas, and Burkholderia. So I'm going to butcher those names. They have also studied the mechanisms of antibiotic and biocide cross-resistance in bacteria. So for example, his research group was able to show a link between triclosan, a biocide widely used in antimicrobial household products, and antibiotic resistance in A. baumannii. His lab is also interested in understanding the role of environmental determinants in antibiotic resistance of A. baumannii and P. aeruginosa. And they have created a number of genetic tools for gram-negative bacteria that allow them to carry out genetic manipulations in bacteria. His lab also studies the prevalence of antibiotic resistance in the environment and have recently shown that drinking water in First Nation communities in Manitoba contains a number of different antibiotic resistance genes, which may pose significant risk to the communities. So we're very happy to have you, and you have the floor.
Thank you very much, Claire, for the kind introduction, and you, Nicole, for the invitation. It's great to be here this morning. Today I'll be telling you a little bit about some of the work we have been doing with First Nation communities, Manitoba, looking at their drinking water. So before I start, I would also like to acknowledge that I also work and live on original lands of Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and the homeland of Métis Nation. And my commitment towards reconciliation is to educate myself about the harms and mistakes of the past, so that I can make meaningful contributions towards reconciliation and collaboration. This is my lab group. This is my lab group from this summer. So I am fortunate to be working with some very smart people in the university, very smart young people, and they challenge me with questions, and they motivate me to find answers for their questions. So when we are here to talk about water today. So when it comes to water, we've all seen that beautiful picture of planet Earth from moon, the blue marble, and yeah, the blue color showing how much water is present on this planet. But there is very small proportion of that water which is actually available as fresh water. So about 3% of all water, and of that 3%, only a third of that is available for our use. So it's about 1% of water that's out there which is available for human consumption. We also rely a lot these days on bottled water, and one thing to keep in mind is that 25% of that bottled water actually comes from municipal water supply. So the role of municipal systems in providing us clean drinking water cannot be overstated. And Claire talked about SDG 6. This is a slide from SDG 6 website. So world's freshwater is or the quality of it is in grave danger. If you look at what we have been doing to our water over the past 300 years, more than 85% of planet's wetlands have been lost when it comes to drinking water. What's going to be, or what's projected for next few years, about 1.6 billion people will lack safely managed drinking water. 2.8 billion people will lack safety managed sanitation, and then also about 2 billion people will be lacking basic hand hygiene facilities by 2030. And this is something to consider, not just from the the need for water perspective, but also from the public health perspective. We have just gone through, as an example, we've just gone through a pandemic, and we know how important hand hygiene is to curb these diseases. So the impact of unsafe water goes well beyond some of the obvious needs that we can think of. Now, when we see these sort of data, we often think about developing countries. Obviously, the problem is much more grave in developing countries. We often, I think in Canada, especially in urban Canada, we don't really see these issues as issues that are directly impacting us. And the reason for that is that in Canada we are very lucky when it comes to fresh water. So if you map our country by rivers and streams and lakes, the country will look blue because we have we have a very good supply of fresh water around, throughout the nation. And some of the work I'm going to be telling you about today is actually from this part of Manitoba. So as you can see that there is no lack of fresh water, but is this water fit for human consumption is what I'll be sort of discussing today. And because we have so much of fresh water at our disposal all around us, we are certainly spoiled as well. So when you look at water consumption around the world and the cost of water, you will see that we by far use most water every day in our lives. So more than 300 liters of water consumption per person. And how much do we pay for our water? We pay actually the least compared to other countries. So you can see that in Germany, water consumption is quite low. And that is probably reflective of the cost of the water, but in Canada, it's the opposite. So again, another reason why when we talk about water, we think the water safety is an issue that doesn't really impact us as much. And when it comes to pure water, there's a tiny village near Toronto that actually claims to have the purest drinking water that's available on this planet, that you'll find anywhere on this planet. So cleanest water, doesn't have heavy metal contamination, doesn't have any source of contamination. So no wonder the quality of our water is considered one of the best around the world. To the extent that next time you go out to buy a bottle water, just look at the source of that water, and you'll see that a number of those manufacturers, what they do is they basically just use municipal water and run it through a couple of filtration cycles and just pack it and sell it back to us. So water for most of us in Canada is quite clean. It's of very good quality. And it is believed that 99% of, when it comes to more than 99% of population in Canada actually has access to clean water. However, there are some exceptions. So drinking water is not as secure of a resource for Indigenous communities in Canada. When we talk about Indigenous communities, 50% of First Nation members live on reserves, and this is data, most recent data that I could download from the federal government website, and it talked about since 2015, so long-term drinking water advisories, and then there are still 28 different drinking water advisories that are out there. Now, again, one of the things to keep in mind is that water, when it comes to water, contaminated water and I'm a microbiologist, so I'm going to talk from microbiology perspective. When it comes to contaminated water, water can contain bacteria, it can contain viruses, and water can be source of various infectious diseases, very serious infectious diseases. And we also know from other parts of the world, access to clean water or lack of it can be directly correlated with life expectancy in various parts of the world. Now, even in Canada, if you look at life expectancy, and this is unfortunately the most recent data I could get from the that we have access to. So life expectancy of the total Canadian population projected to 79 years for men and 83 years for women. However, when you look at Inuit population, for example, the life expectancy was 15 years less for men and about 10 years less for women than total Canadian population. So there are various factors that contribute to this. but infectious diseases has to be a part of it as well. And then when we talk about infectious diseases, and I say infectious diseases because we know that, for example, rates of tuberculosis in Inuit population are 300 times higher than rest of Canada. So infectious diseases certainly are contributing to this. And when we talk about infectious diseases, you know, you could assume that water has a role to play in there as well, although we don't really have a clear-cut data set to show that. So when we talk about drinking water advisories, there are three kinds of drinking water advisories. So there's a boil water advisory where you have to boil your water. There is do not consume advisory where you should not be drinking that water at all. And then there is do not use advisory, which basically means stay away from that water. And you know, as I said, that there are communities out there which have one or more of these advisories ongoing at the time. And this is a picture that was taken by one of our graduate students who was visiting a community, and here you see a do not consume advisory, basically do not drink that water. So when these advisories are in place for more than a year, that's when these are defined as long-term drinking water advisories. So this is something I would like you to sort of remember throughout this talk, long-term drinking water advisory, because this is where we have most focus on right now from the government side of things, looking at ending long-term drinking water advisories. So this data is actually, again, this is most recent data that I accessed last week. And when you look at this is looking at long-term drinking water advisories across the country. You look at these numbers, 5 remaining in Saskatchewan, 3 in Manitoba, and 20 in Ontario. So that's about 28 drinking water advisory, which is the number I showed you previously as well. And you know, from long-term drinking water advisories. So from 2015, when there were more than 120 different drinking water advisories, it seems like down to 28, which is great. And but what I'm going to be telling you about is that this is not the complete picture when it comes to the quality of, or the lack of it, of drinking water in First Nation communities. So drinking water advisories do not necessarily provide full assessment of the water quality in these communities. So, for example, a number of these communities actually rely on private water supplies, wells, cisterns, and homes, and homes without water service. So, how is the water quality in these households monitored? And there is no system in place actually to monitor that for these households. So what we did was for our work, this work that I'm going to be telling you about, we went to some of these communities. We worked with some of these communities, and what we did was we actually sampled their water. Now, the communities we went to work with, none of these communities were under drinking water advisory and long-term drinking water advisory. These communities also have water treatment plant, which is one of the foci of federal government trying to put in water treatment plant was in place to ensure safe drinking water for community members. So they had water treatment plant, and then when it comes to the water distribution system, they had sort of a mix of system in place. So they have some households have pipelines, so they get water directly from treatment plant. Some households actually rely on water trucks bring in water to the cisterns in the household, and these cisterns can be made of different materials. So some made of plastic, some made of cement. And some of these communities were fly-in communities, so that that did add to the challenge in terms of sampling. When we are looking at microbiological quality of any sort of sample, the time between sample collection and analysis is very, very critical, because bacteria tend to grow and they grow fast. So if there is a delay in sampling, then what that can do is that can lead to inflated number of bacterial cells, because they will divide, multiply, and then give you higher number in your samples. So this is a picture taken by one of our students who was going to one of the communities for water sampling. And for sampling, obviously there were community members who were helping us out. They were very gracious in helping us out. So this is collecting samples from water treatment plant, and then often, so this is the group of students who went out for sampling, and then this is a community standpipe collecting sample from there. And then often, young children from communities will come along and see what the students were doing, how they were collecting samples, and what they were measuring. So first thing we did was we actually looked at microbiological quality of water. So this is, when it comes to microbiological quality of water, one of the things, the most critical marker that we look for is total coliforms and E. coli. And for non-microbiologists in the audience here, coliforms are the organisms that are released with fecal matter. So they come out in our feces. Coliforms do not have to be pathogens. They do not have to, because our gut contains a lot of bacteria, and with every bowel movement, we release a very large number of bacteria in billions into the environment. Now, because they are released in such high number, they're easy to detect. So if there is fecal contamination of water or fecal contamination of any material, for example, you would, in order to detect that fecal contamination, you would be looking for coliforms. So these are the markers of fecal contamination. Doesn't necessarily mean that they are pathogens or disease-causing organism, but if there is fecal contamination, it's very likely that they are the sample is also carrying pathogenic organisms, because anybody who's sick will be releasing pathogenic bacteria, disease-causing bacteria in the environment as well. So we look for coliforms and we look for E. coli, which is one of the coliforms. E. coli is an organism, once again, it resides in our gut. Most strains of E. coli are not pathogenic, but there are some nasty ones that can make us quite sick. So what we did was we looked for these two organisms to analyze for fecal contamination in the water samples. So this is basically how we would look at. So you see these dots. These are E. coli and other bacterium growing on agar plate. So these samples were collected. Water samples brought to our lab in Winnipeg, and then they were basically analyzed for presence of bacteria. So these, when we look at total coliform, for example, so basically just look at the bar graph. Now, the guidelines say that drinking water, to be safe for drinking, should not have a single coliform or E. coli. So 0. 0 is the number that we are looking for in order to make sure to ensure that the water is safe for drinking. But here what you can see is, and I won't worry too much about these notations here, but these are tap water samples, there are cistern samples, and then these are lake water sample, and these are samples that we used from tap water in Winnipeg as our control samples. So what you can see is that tap water samples have some coliform, so it should be 0. There is some coliform, but when water gets into cisterns or in buckets in household, then we start seeing high number of coliform. These two samples right here are the samples collected directly from the water treatment plant in these communities. And you see that there is absolutely no coliforms present here, no E. coli present here as well, which tells us that the water treatment plant is working exactly the way it should work. It's cleaning out all bacterial contamination. But when the water gets into the distribution system, for some reason, these bacteria are coming back into the water. So once again, just keep in mind is that this particular community was not under any water advisory. And the reason is because their treatment plant is working perfectly fine. But then because this is not under water advisory, we are assuming that everybody has access to safe water. And obviously, when we look at this data, that is not true because people are getting exposed to coliforms and E. coli. So then what we did next was we actually did something known as microbial community analysis. And this is, again, for non-microbiologists in the audience. This is simply a fingerprinting analysis, if you may, looking at what kind of microbes are present. So the idea is very similar to how, DNA fingerprinting is done. How, when we do DNA fingerprinting, you can find somebody's DNA, you read the DNA, and then you match it with the person whose DNA it might be. And that's how you can find out whose DNA was it. So this is what we did here. We extracted DNA, we read DNA, and then we found out, we matched that DNA with different kinds of bacteria just to see what kind of bacteria are present in these water samples. And one thing to, again, remember is that bacteria are present all the time, and presence of bacteria is not a problem because most bacteria are good for us or do not cause us any harm. So finding bacteria is not a concern. However, finding bad bacteria is a concern. So here again, this is a busy slide, but what I'm trying to show you here is if you just pay attention to the color pattern. So this color pattern basically shows what different kind of bacteria were present in the samples that we tested. So basically, if you look at the lake water samples, this is water sample that was before treatment. You see that there is a lot of blue here. And this blue is actually representative of bacteria that are coliforms, E. coli, and other related organisms. So that is telling you that there is probably fecal contamination in there. Then you run that water through the water treatment plant, and you see the microbial profile actually changes, the community profile changes. So now you have other organisms which are actually harmless that show up. So we find those DNA there. But then when the water gets back into the distribution system right here, you see that it starts representing the lake water samples again. So very similar to lake water samples. So obviously something is changing after treatment, which is making water contaminated again, or the microbial profile is now same as untreated water. So this data set actually gave us a good sense of what might be, well, how that is happening, we don't know, but what might be happening here that, water gets treated, bacteria get removed, and then there is recontamination of water when it's distributed to households. So, when we found all these bacteria, different kind of bacteria in water samples, then we were interested in looking at antibiotic resistance. The reason for antibiotic resistance is that, resistance is when antibiotic resistance is a phenomena where water sample, where bacteria that cause infections in human can no longer be treated with antibiotics. And that is a serious problem because that basically means that we are unable to treat patients who have bacterial infections. And antibiotic resistance comes from various sources. It can come from hospitals. It can come from agriculture setting. It can come from our households. But it all seems to get to water, right, in our environment. So this was the reason when we started finding bacteria in these water samples. We want to see if bacteria that are present in this water are also resistant to antibiotics. So before I show you some of that data, I just want to tell you a little bit about antibiotics and modern medicine. So antibiotics are used to treat bacterial infections, but antibiotics have also contributed a lot to modern medicine in general, right? So when it comes to antibiotics, surgeries, the kind of surgeries that we we can carry out today wouldn't have been possible without antibiotics, because rate of infection in these surgical procedures were very, very high before the use of antibiotics. But now we are able to do that without getting infected. When it comes to treating chronic conditions such as kidney disease, again, antibiotics prevent infection. Diabetes, cancer treatment, antibiotics are needed to prevent infection. Organ transplants, we won't be able to do organ transplants without antibiotics. So think of hip replacement surgery, think of knee replacement surgery. None of that would be, or those would be very, very high risk procedures if antibiotics were not there. And then cancer care would be also almost impossible to carry out without antibiotics. So it is believed that antibiotics have added 20 years to each person's life today. Now, these numbers are just, I'm showing you this number here, that if you can treat infections, then we can live 20 years more than individuals where we cannot treat infections. But I also want you to think about that life expectancy data I showed you in the beginning of this presentation. So is there a link between antibiotic resistant infections versus life expectancy? We don't know right now. We don't have any data to show that, to prove that, but some of these numbers are interesting. And it is believed that antibiotic resistance is going to be, if nothing is done today, by 2050, they will be killing about 10 million people around the world every year, which is actually cancer and road accidents combined today. So very high number of mortality from antibiotic resistant infections. In Canada too, it is believed that about a quarter of the infections that we see are resistant to antibiotics, which means that those are difficult to treat. So resistance means that people will get infected, and then we would have problems treating those individuals, which means that it will add to the cost of healthcare. It would actually add to the mortality of individuals. It would result in more specialized hospital facilities needed. So the impact of resistance is very, very widespread when it comes to health of the community and healthcare system. So what we did was we looked at antibiotic resistance genes in these water samples, and we actually found that this is one of the genes that we looked at. This gene is responsible, or the gene product is responsible for resistance to penicillin group of antibiotics. And we found that some of these water samples that we had actually did have antibiotic resistant genes as well. This is a gene product that causes resistance to tetracycline group of antibiotics. And once again, you can see that this was quite prominent in every single water sample, except for the water sample that was collected right at the plant after treatment. So once again, this is telling you that the water treatment plant is working perfectly fine. And that's why these communities are not on anyone's radar when it comes to the lack of drinking water quality. Whereas, and we see Winnipeg tap water also did not have any of these genes. And MEC-A, some of you might have heard of a strain of Staphylococcus called MRSA, or methicillin-resistant Staph aureus. MRSA causes quite serious infections because it cannot be treated with most antibiotics that are out there at at our disposal. And we found once again MEC-A gene, so potentially MRSA, in these water samples. So this is basically telling us that antibiotic resistant genes are present in these water samples. We went back to these communities and we wanted to look at if there is a seasonal effect on the number of bacteria on these in these water samples. And the take-home message from this is that we didn't really see too much of seasonal effect. We did see higher numbers in August and October when the water is warm. So, but But what we saw was actually that it was the cistern water samples which tend to have higher number of bacteria. So among distribution system, it's when people are storing their water, particularly cement cistern, we see high number of bacteria. And that's the same kind of pattern we saw in another community. This community actually didn't even have cement cistern, they had plastic cistern. And once again, it's showing high number of bacteria in cistern samples. So what this is telling us is that we need to have a policy in place to ensure that these cisterns are cleaned in a timely manner. We also looked at other organisms. So we looked at E. coli and coliforms, but then we wanted to see if there are other pathogenic bacteria present in water as well. And the organism we looked at is called Campylobacter. Campylobacter causes some serious GI tract infections in North America. Primarily comes from eating raw or undercooked poultry. But when we looked at the presence of Campylobacter in our water samples, we actually found that, yes, Campylobacter genes, we didn't look for the organism, we looked for the genes. So same idea as DNA fingerprinting, but we found that Campylobacter was present in these water samples. And what was alarming was that Campylobacter genes were detected even in water samples that did not have E. coli and coliforms. So it seems like the problem is way beyond just E. coli and coliforms as well. And we saw a similar sort of pattern when we looked at antibiotic resistance genes in these samples. Once again, cistern samples had higher number of resistant genes. So what we are finding here is that when we have, and this is data from the other community, that when water, if we have to target one aspect of water distribution system, or if we have to prioritize one aspect of water distribution system, we have to look very carefully at water storage in these systems. It seems like there is a tremendous health risk associated with the cistern water samples. So what we wanted to do next was, once again, just looking at sort of a DNA profiling of these water samples. We saw high number of resistant genes and bacteria in cistern samples. So we wanted to just make sure, compare the microbial profile of source water with cistern water. So the idea was that, okay, water treatment plant is working fine. It takes away all the bacteria, but then all the bad bacteria, and then water in the cistern gets bacteria back again. Are these bacteria coming back somehow from the source water? So what we did was we compared the microbial profile of source water, untreated source water, with cistern water. And without going into the detail, if you look at this heat map, these are the source water samples, and this is the cistern water sample from one community. And you can see that the profile is very different. So this tells us that the contamination of this cistern sample is not from the untreated water. It's some other source. This is true for the other community as well. This is the cistern water. You can see that the profile is quite different from the source water. So the contamination, the source of contamination is something else. When we looked at resistance genes, same sort of pattern, that the pattern was quite different from what we see in the source water. So it wasn't the untreated water that was contaminating, or at least from what we can tell from this small pilot study, that was contaminating these cistern water samples. There's something else going on. And when we did, when we looked at basically just the correlation, that's exactly what we found. So these are the source water samples from two communities. These are color coded. So one community here, one community here. This is Winnipeg water sample. So source water samples are, you can see that they cluster together, but this is the cistern water samples. And this is looking at antimicrobial resistance gene profile, and also we looked at the microbial profile. So very different profile, which basically means that the source of contamination is different as well. So what we have been doing is going back to the community and sharing our data with them. We provide them what we find, and then we have also had gatherings where community members were invited, and then we would share data with them. This was all pre-COVID. We haven't been able to go back to the community since COVID, but we have tried doing this, and this is something that I realize we can do better in future as well, engaging communities more with our analysis. So fine, with that, I'll just end here. So poor quality of water, it's a problem when it comes to AMR in antimicrobial resistance in First Nation communities. What we are finding is that systems are probably the biggest health risk when it comes to water, access to safe water. And this is something that needs to be addressed. Again, keep in mind, neither of these communities were under any kind of drinking water advisory, but we are finding that people in households are at tremendous risk when it comes to bacterial infection. So these are the students from the lab who actually carried out all the work. This is during one of their trips. My collaborator in this is Dr. Annemieke Ferenhorst, who actually introduced me to this project, and we have been working together ever since. And then obviously wouldn't have been possible without the help from the community and community members. So with that, I will end here. I will just leave you with this particular slide to sort of think a little bit about. We always talk about lack of funds for ensuring safe drinking water for people around the world, people in Canada. But there is money around there. Anyways, with that, I'll end, and thank you so much for your attention.
Thank you so much, Ayush. That was really, really eye-opening and interesting. So we have time for questions. I see that. Oh, go on. And I forgot to mention, we do have a Q&A as well as a chat, so feel free to post them in either. I see in the Q&A, we have one. I'll read it. So it says, wow, I can empathize that the lifting of a water advisory might logically cause a person to think that water is potable. So thanks very much for digging deeper into this water quality issue. And yeah, that was one of the things I took away from your talk straight away. Water quality seems to be a splendid way to blend the learning required in a variety of disciplines, such as engineering, basic science, microbiology, of course, any courses in which social issues of contemporary is a fundamental hallmark, equity issues, and on and on. Do you see it as possible to use issues like water quality in a wider variety of courses? And if so, sorry,
I'll finish the questions. Sorry.
Yep. Oh, so most of that was a comment. The first question I see is, do you see it as possible to use issues like water quality in a wider variety of courses?
In courses?
If so, yeah. And if so, what is the key, the missing link to ensuring that rural issues such as drinking water quality are able to be more prominently used as learning context in higher education systems?
Yeah, and that's a very good question. Thanks for that question. So I'll give you my example, right? I teach a course in microbial diseases, and before I got involved with this project, so this was about more than 10 years ago, I had inherited this course from a colleague of mine, and we would talk about water quality, but there was no mention of water quality or lack of it, microbiological quality from First Nation communities in Canada. So there were a lot of examples from rest of the world, and honestly, when I took up that course, I didn't know about that either. And then as I got involved, and as I learned more and more from gatherings like this, I realized that there are various instances just in my course where I can actually talk about some of these issues, and I do now. So I think the key thing is, because often when we talk about courses, one of the and it's a normal reaction. I think a lot of times people say, well, this is an important issue, but I'm not really sure if it applies to my course or what I teach or the learning objectives in my course. And that's where I think we can probably, so rather than just even thinking about, hey, you know what, maybe you should talk about this. I think what is important is to give a better sense of the context here. And then I think, because I can see something like this in a microbiology course, in actually a chemistry course with, I didn't talk about some of the chemical analysis we have done, but that aspect is there. A course in social sciences, I would see that, so it's just, how do we ensure that people get the context of these issues that they feel that they can apply in their courses? And that's, I think, so the discussion is, has to be, maybe we have to take a step back and sort of start thinking about that.
Great, yeah, thank you. Okay, so another question is, what percentage of the drinking water in this, I think it says Lion, I'm not supposed to be sure if it's Lion community, is distributed through cisterns, and especially concrete cisterns?
So it varies. Some communities, they were actually very, I would say, a large proportion of cisterns were concrete cisterns. With one particular community, we actually, when we shared our data with them, they were able to get some funding to replace their cisterns. There was one community, fly-in community, where they have absolutely no cement cisterns. So there is sort of an effort to, any replacement of cisterns will not be with cement cisterns. So plastic cisterns are coming in, but and some fiberglass cisterns as well. But what we are finding is that, I think the key thing is, well, cistern is obviously a problem for cement is obviously a problem for various reasons, including cracking and so on and so forth. But even with plastic cisterns and fiberglass cisterns, if we're not maintaining them properly, then bacteria will start growing in there. So just replacing cisterns is definitely part of the solution, but I think for the solution to be sustainable, we need to come up with this with some sort of policy where people are and resources where people can get those cisterns cleaned on a regular basis.
Cool. And I'm just going to add to that question. So, are there documented? I mean, so obviously they sell, or cisterns have been around for a long time. So, are there current methods that you know of that are effective in cleaning, say, plastic cisterns for communities? And do you have any sense of the costs that would be associated with that?
So I don't have a good sense of the cost. I do know that resources tend to be lacking. So, for some of the communities, if there is one person who's sort of responsible for cleaning these cisterns, they can't even get through all the cisterns to community in 1 year. So that we know. I mean, when it comes to cleaning, the idea would be, the simplest way to clean those would be, shocking them with chlorine. However, with cement, there is a problem because cement the surface is uneven. So bacteria are forming biofilm, which basically means aggregates, large aggregates, and those can be resistant to, so you actually have to have a fair bit of scrubbing as well. And that adds to the time and the cost and the resources. And in terms of proportion of cisterns, cement cistern, that can vary a lot from community to community.
Interesting, thank you. Okay, so here's another one. So Steph is interested in your risk communication strategies. So in addition to your really useful community presentations, what about complementary approaches, workshops, and videos through social media, as well as infographics, both around the bad news, but also the good news that the treated water is safely piped in, for example? My experience, so this is from Steph McLaughlin, my experience is that many or most community members don't trust the locally treated water, and so spend a lot of money buying bottled water, which, as you say, is often pretty much tap water anyway.
Yeah, so Steph, that's a wonderful suggestion. And honestly, until I heard about your videos, some of the videos you have created, I hadn't thought about it, but this is something that we would definitely like to do. Actually, I'm going to pick your brains on this a little bit as well. because that's certainly a strategy that we need to employ. So use of social media and videos actually. Social media is on personal front of I guess on personal front, I'm just getting off all social media platforms now, but I know that people do find those useful. But videos, making videos and animations is something that we definitely want to do, Steph. So that's something that I'm going to have a discussion with you.
Neat. Okay. So this one's from Ricardo. Great presentation and message. Absolutely. One curiosity, can the DNA in the bacteria in the distribution system allow you to track its origin?
You know, Ricardo, it can. We just haven't done the study of that scale. We were able to rule out source water. Last few slides, that the data I showed you on last few slides. But where else it is coming from, it is certainly possible. We just haven't been able to do it yet.
Cool. And then there's a comment from Miguel. Freeze thaw in these areas impact on the water reservoirs or cisterns, combined with seepage filtration from septic tanks. Bacteroides is an indicator of human fecal contamination. And great presentation.
So Miguel, we did actually look at, I didn't show that data, but we did have there was a student who actually looked at potential of septic tank contamination, and we did not see that. This was in one community and few households. So not to say that, again, you know, the water distribution system, as you know, you all know, is it varies a lot from community to community, but at least in one community, when we looked at it, we did not see any evidence of contamination from septic tanks.
Interesting. And this is from Eric. I've heard a lot of complaints about the chlorine taste in the drinking water from the treatment plants. Is this one reason people use holding tanks to let it outgas? And what do government officials say about the issue of people not actually using the treated water for various reasons?
Well, people are using treated water. It's just the issue is that, so the water that is coming to their cisterns is treated and is coming through the trucks because they don't have access to pipeline. You know, they don't, the households are not piped. So that is certainly, so the water is being treated. It's just getting recontaminated. Now, in terms of chlorine, yes, the smell of chlorine and the taste of chlorine can be quite you know, it's not pleasant at all. I don't think we find it pleasant, but I don't know anyone who finds it pleasant, but it's just, you know, when you have such high number, one of the challenges with chlorine also is, and I didn't show that data, but we also looked at chlorine concentration. And when the contamination goes up, chlorine concentration goes down, or vice versa. When chlorine concentration goes down, contamination goes up in these cisterns. And that could be because of organic load too. So if there are organic compounds present in there, they tend to dissipate chlorine. So it is not as simple as just adding chlorine to the water, because if the cleaning is not proper and if the organic load doesn't go down, then the chlorine, you know, it's tough to maintain that desired concentration of chlorine.
Okay. Thank you. Here's a question. I believe it was in the chat. Water quality seems to be a splendid way to facilitate learning in a variety of disciplines, such as engineering and basic science, as well as the social issues of contemporary life in our local areas. I think that's just a comment. And there's another question from Nicole. She's curious about the potential to use technologies for treatment at source like UV tech. Have you looked into this as an option?
So Nicole, that's something that we want to look at. We haven't looked at it yet, but this is something that we are sort of starting to wanting to look at. That's one for sure. I think UV systems work quite well in seen evidence from some of the developing countries. They work quite well, but we haven't tried those here. Something that we would like to try in future.
So this is from me. So it sounds like there's a lot of work that can still be done. And I know you mentioned going back into the community and building communication tools as well. But what do you see as your next steps, if you can? Like, would you be able to continue this research and expand on it?
We would certainly like to continue. And I think the next step is actually, so two things. One thing is that what Nicole mentioned in terms of the solution. So we would like to explore UV treatment options in household settings. And the other thing is what Steph said, actually, outreach, increasing our outreach activities. I think when it comes to surveillance, it is important, but based on what we have seen in few communities, I wouldn't be surprised if this is what we see in most of the communities, if not all. I mean, there are a few communities that we went to where water was quite clean, but most of the communities, I think this problem is, I won't be surprised if it's, it exists in most of the communities. So the next step is certainly looking at what are the potential solutions and also informing people about, goods and bads of what we are finding.
Interesting. Okay. And this question's from me. So I noticed in one of your slides when you were breaking down, I don't know if it was the genuses, you had like oscillatorials among them. So to me, of course, being a algal person, oscillatorials are also algal species. So are those, for some reason, do you have a whole range, like a specific range of genus that you look at that include algal species? So for example, I'm thinking cyanotoxins that probably aren't routinely monitored right now either.
Yeah, we did not look at cyanotoxin, for example, but this was basically, so this was just 16S sequencing. So just looking at a small region in the DNA and just trying to match it with, so we just ran the sequencing and then match it with what's in the database, and that these are the results we were finding. So there was a mix of bacteria and other organisms as well, and that's why I think it was showing up there. And probably it's there, that's why it's showing up.
Great, thank you. And Adele says, thank you for the presentation. It was such important research, or it is such important research.
Thank you.
And that brings us pretty much to almost 12:30. That was a really great presentation. Does anybody have any last comments or questions? Otherwise, you will be able to see this presentation on our website, hopefully within the next week or so. And I want to thank you again for taking the time to present this to us. I learned a lot. And thank you everyone for attending our first seminar series for the 2023-2024 season. We have 5 more talks. Next month on October 23rd, it will be Amy Crafts speaking about building legal relationships of responsibility to and with NIBI, which is water. So have a great rest of your week, everyone. An amazing job, Ayush.
Thank you very much.
Bye.